Hello, my name is Ariana Roth. I'm a third-year student at the University of Wyoming studying chemical engineering with a minor in biomedical engineering, and I work in Dr. Oki's bioengineering research lab on campus.
Hi, my name is Andrea and I'm a junior at the University of Wyoming. My major is physiology and I work in Dr. Oki's lab. And before we get started, we just wanted to say a huge thank you to the Wyoming Research Scholars Program for putting on this symposium. And this is the fabrication of microfluidic devices to optimize experiment environments. Microfluidics have a vast array of applications in bioengineering. Specifically in the Oki lab, we deal with microfluidic devices. Microfluidic devices are composed of polydimethylsiloxane, or PDMS, which is a silicone-based organic polymer. Each one of these devices contain a unique pathway, which is specific to the function of the device, along with various inlets and outlets which are used for pumping desired solutions into and out of the device. Microfluidic devices are incredibly versatile and create a controlled environment for various experiments. Additionally, these devices are relatively inexpensive to produce and preferable to large-scale counterparts. Thus, they are an ideal interface for biological and medical experiments involving rapid diagnostics, live cell imaging, and drug research. Microfluidics even have various technological applications.
The most common material that we use in our lab to make microfluidic devices is polydimethylsiloxane, or PDMS as it is commonly referred to. PDMS is a very common material in general to fabricate microfluidic devices as it is transparent, readily available, relatively easy to work with, easy to obtain, and economical. Another material that we use in the lab to make the devices is polyethylene terephthalate, also known as PET. Microfluidic devices can be made with different materials, and the material used depends on the application of the devices and the certain experiments they will be used in. PET is more chemically resistant than PDMS, therefore microfluidic device material can be chosen to suit the experimental requirements. Another type of material that we use in the lab to make devices is polyurethane, which is helpful when designing a device for cultivating cell cultures. The first step to making a microfluidic device is having a device design. These are made on the computer on a software such as AutoCAD. This is the first step to fabricating specialized devices and manipulating the depth of the fluidic chambers to fit specific experimental designs. The design is then printed to make a photomask, which can be used over and over again to make more wafers. A silicon wafer is fabricated with the specific device design using a technique called photolithography. This is a lengthy process and can take anywhere between 40 minutes and a couple of hours. Photolithography uses a process of using a photoresist, an SU8, and depending on the depth of the chambers of the device one is trying to obtain, a thicker or thinner photoresist can be used. The wafer is first cleaned using pressurized air, heat, and placed in a plasma cleaner. Once the silicon wafer is cleaned, the photoresist is placed on the wafer, and the wafer is placed in a spin coater apparatus. The spin coater ensures an even coating of the photoresist across the surface of the wafer. After that, the wafer is heated by ramping for a slow evaporation of the solvent and helps to ensure that the other chemicals can spread more evenly on the surface of the wafer. The wafer is now ready to be imprinted with the device design. Wafer is placed under UV light and exposed for a time determined by exposure energy divided by the light intensity with the mask on top of the wafer. The wafer is again heated and then placed in a developing solution and left to sit for a few minutes. The wafer is then cleaned and can be ready to be used to make PDMS devices. Once the wafer is finished, it, like the mask, can be used over and over again to make devices. The wafer is taped in the bottom of a petri dish, and the PDMS devices are cut out of the petri dish. After a wafer is successfully made, it can take a couple of attempts. Photolithography is a very complicated and detailed process, and the petri dish is filled with a mixture of PDMS and curing agent in a 10-to-1 ratio. The PDMS and curing agent is weighed out, and usually to fill a petri dish for the first time, it is around 70 grams of PDMS. The petri dish is then placed in a vacuum chamber to suck all of the air bubbles out of the PDMS for about 20 minutes. Once the air bubbles are out of the PDMS, it can be left to set. It can be set at room temperature, and that takes about 4 days, or it can be placed in the hot oven at about 100 degrees Celsius if it is in a glass petri dish. and that will take about 10 minutes. And if it's in a plastic petri dish, it's placed in the dryer where the PDMS sets in about 3 to 4 hours. Once the PDMS is set, it can remain in the dryer or at room temperature until one is ready to cut out the devices. The devices can be cut out of the petri dish using a scalpel, and inlets and outlets are also punched into the PDMS so that fluids can be placed in the devices to run experiments. There's only one step left before we have a completed PDMS device, bonding to a glass slide or cover slip. The glass is cleaned with ethanol and plasma cleaner, and the PDMS is also cleaned. Then the PDMS is placed in the plasma cleaner with the glass for about 40 to 45 seconds. This changes the surface chemistry of the PDMS so that the PDMS and the glass can become chemically bonded. The imprint side of the PDMS is bonded with the glass, and now you have a finished device. The device is ready to be used in an experiment.
One of the most medically relevant applications of microfluidic devices involves a protein involved in blood clotting, fibrin. Fibrin is produced from fibrinogen, which is found in blood plasma. When the enzyme thrombin acts on fibrinogen, fibrin is rapidly polymerized, causing the reaction of a mesh-like structure, which, in conjunction to platelets, causes a clot. As seen in the image to the left, fibrin has a multitude of clinical applications, but one of the limits of fibrin in a therapeutic setting is that it has a relatively short degradation period of a few days. Degradation can be reduced by increasing the concentration of the fibrin in the solution. So how do we produce these droplets using microfluidic devices? To begin, the device is customized to produce droplets of a specific size and composition. In our lab, we commonly use droplet generating devices that produce droplets that are 50 microns in diameter. To begin, the desired solutions are prepared. In this case, a solution of fibrin and oil are needed. The fibrin solution is placed in a vial and pumped into the microfluidic device via inlets. And this is pictured by the image on the left, and the inlets are the two entrances into the device on the left of this image. After entering the device, the oil splits into two directions and meets up with the fibrin solution at an intersection, pictured by the images on the right. As both the oil and fibrin solution are pumped, droplets of aqueous fibrin are created. The formed droplets are guided to the outlet, which leads to another beaker containing droplets. These droplets will later be polymerized. These droplets can be used in various in vivo situations, most notably in regenerative medicine as degradable cell scaffolds during drug delivery or in reducing scar tissue formation in wounds. Another application of these microfluidic devices is live cell imaging, which is extremely useful in the biomedical field when it comes to cancer and pathogenic research. Microfluidic devices can be used to simulate an in vivo environment within the device, which reduces the need for animal testing. The devices are referred to as organs on chips. Specifically, in the study of cancers, other in vitro cancer models are insufficient in creating the exact microenvironment in which the tumor forms. These chips allow scientists to vary cellular, chemical, molecular, and biophysical parameters in a controlled manner. For example, a lung on a chip is pictured above. The model contains human epithelium and endothelium cells and a porous membrane between them. Air flows across the top portion of the device and blood cells along the bottom portion, mimicking the function of human lung cells. Additionally, a vacuum creates a pulsating sensation along the side chambers, which again mimics the actual environment of lung cells. Various pathogens and cancer cells can then be passed over the lung cells, causing infection. The mode of infection can then be studied and observed. The organ on a chip model also acts as effective devices for drug testing.
A final application of microfluidic devices is both COVID-19 research and COVID-19 rapid testing. In our lab, we are developing a prototype of microfluidic devices made out of PET to be used for COVID-19 rapid testing as another way to test for the viral proteins in the saliva. These PET devices are made from the Epilogue laser printer in the makerspace here on campus, where small pieces of PET and the adhesive are cut from the laser, and then chambers of the device can be made by sticking the PET pieces together using the adhesive. These devices are still being developed and tested. We are using PET films for these devices because they are inexpensive, durable, and flexible. With PDMS, we use glass slides, which are fragile. Capillary action from the microchannels will draw in the fluid sample from the person being tested. In the device, there will be functionalized hydrogel posts that will catch specific proteins correlated with the virus and signal a positive fluid sample. In conclusion, microfluidics can be used for a wide variety of research experiments in biomedical fields specifically. There are different materials like PDMS and PET, as we talked about, and different fabrication methods to accommodate the requirements of specific experiments.
Finally, we just want to say thank you again to the Wyoming Research Scholars Program, to our PI, Dr. Oki, and to Cassidy Enlow, who works very closely with us in the lab.